Okay, hi, I'm Steve Angeloni, a senior field application scientist at Luminex Corporation. In this presentation today, tools for calculating bead concentrations and making multiplex bead mixes, we're going to cover the following topics. When do you need to determine bead concentrations? We're going to look at methods for determining bead concentrations after covalently coupling molecules to beads or binding them to Mag-Avidin tag beads. We're also going to look at calculations for determining concentration, percent recovery, and making coupled bead stock dilutions. And then we're going to take that information and learn how to do calculations for making multiplex bead mixes. So let's start with when do you need to determine bead concentrations? Well, the first thing I can say is there's one time where you will not need to determine bead concentrations. And this is when you're using our X-tag beads for development of genomic applications. The bead concentrations for these beads come in the product specification sheets that are shipped with each vial of beads. In general, however, you will need to know bead concentrations after you've coupled proteins, oligos, or other capture molecules to our Luminex beads. If you're using biotin-tagged proteins or peptides that are being bound to Avidin beads, you'll also need to know those concentrations as well after you've done the binding of your conjugated molecules to the Avidin-coated beads. So what's the importance of knowing bead concentrations? Well, because of bead loss during the coupling or binding process, there can be a significant decrease in the amount of beads being recovered. So not knowing how many beads are actually recovered and added to assay reactions can result in significant performance differences due to variations in bead concentrations in the assays. So knowing the percent of beads that are recovered after coupling or binding to the Avidin beads can also let you know if there are any problems with your coupling and binding procedures. So knowing the bead recovery concentrations is an important QC step when developing an assay and ensuring consistent performance across different lots of captured bead preparations. So let's look at some of the different methods for determining bead concentrations after coupling or binding to Avidin beads. So one of the basic techniques that we'd like to teach people is how to use a microscope and hemocytometer. So methods on how to use a microscope and hemocytometer in the XMAP cookbook. Although this method is the most cost-effective way to determine bead concentrations, it can be very time-consuming when you're developing high-plex assays, and it may be subject to variation with different users. So another approach is to use a cell or particle counter. There's a number of these available. So counters can be quick, and depending on the instrument used, will allow the user to save details of the bead preparation, like the bead concentration, percent of clumping, the average bead size, and other information. They are also less prone to counting variation by different users. So there are a number of different automated counters available that have been used for counting XMAC beads. A number of them are listed here. These include something called Luna counter that's available through the manufacturer called Logos. Bio-Rad also has an automated cell counter called the T720. Beckman Coulter has a high-end counter called the multi-sizer for E Coulter particle counter or analyzer. Thermo Fisher LifeTech also has something called the Countess. There's also several counters from Nexalon Biosciences, as well as Millipore has a sort of pipette style counter called the Scepter 2.0 cell counter. So features you want to look for in a cell counter, if you're going to choose one of these, include the following. First of all, you want to make sure that the counter requires a small sample volume of your bead preparation, preferably a volume of not more than 10 microliters. You also want to make sure that it can measure particles in the size range of 6.5 to 5.6 microns. This will enable you to count accurately either microplex or magplex beads if you're using either of these in your assays. You also want to make sure that the counter can give you accurate counting in the absence of trypan blue. There are some counters that default to a trypan blue compensation, and if you cannot turn that off, it will give you incorrect cell counts, or bead counts in this case. So you also want the counter to have the ability to generate reports that show information like the bead concentration, the average bead size, the percent clumping, and maybe even give you bead images and other information. So whichever counter you select, you should make sure that you can program and calibrate the counter with uncoupled beads from stocks that you can get from Luminex. So the actual concentration every stock is on the specification sheet that is shipped with every vial. And the counter should let you set the parameters so that you can get as close to that concentration as possible. So let's look at calculations for determining bead concentration, percent recovery, and looking at how to dilute our coupled bead stocks to a uniform dilution. So in order to do these calculations, there's an Excel workbook available that has 2 tabs in it. The first tab lets you pick a volume that you want to make for each bead stock and the concentration you want that bead stock at. The second tab lets you take a whole coupled bead stock and dilute it to a given concentration so that all your bead stocks will have the same concentration. So let's see how this spreadsheet works. So let me minimize this a little bit here so you can see more of the screen. And let's say we have 5 different bead preparations, and we're going to call these just protein 1 to 5. So that's going to be our protein 1. And let's make 4 more. I'm pulling them down. And this one's going to be on bead region 42. Let's put this on bead region 34. This can be bead region 35. That can stay at 45, and this can be bead region 26. So let's say we coupled our beads so that we coupled them at, say, 4 million beads total, and we resuspended them in 500 microliters. That would give us an expected concentration of 8 million beads per ml if we recovered all those beads back. And let's say that when we needed to count our beads in our cell counter, or our automated counter, we had to do a 2-fold dilution. And when we got those counts back from the counter, one of the counts, let's say for protein 1, came out to this concentration. The other for protein 2 came out to this concentration. This one came out to here. And let's say this one came out a little bit lower to, let's say, 3,600,000. Is that one more 0? Yep. And then let's say this one came out a little lower. Maybe this was 3,580,000. Okay, so with these different concentrations at a 2-fold dilution, the actual concentrations of these beads is going to be this amount per ml. And let's say for each of these, we want to make 1 ml of that bead stock, and we want them all to be at 1 million per ml. So what these volumes tell us here is that it says that we're going to need this much of each bead stock plus this much of an assay buffer to give us the 1 ml, so that each of them are now at 1 million beads per ml. The other thing this spreadsheet shows us is it shows us our percent recovery. So there's very little loss here. Most of the recovery is about 90% or more. This one's a little on the low side, but it's still not that bad. This would still be usable in a Luminex assay. If you start getting percentages up down into the 70%, that could be a little more concerning, and you may want to recouple those beads if they don't perform well. In any event, remember, we were using this example where we coupled 4 million beads and we brought them up into 500 microliters. So each time we want to make, say, 1 ml of them at 1 million beads per ml, we have to take this much out of each bead stock. And over time, you may lose track of how much has been taken out of that 500 microliters. You know, maybe there was 10 microliters taken out to do your calculation on the counter. Then you're taking out this much, or maybe a different amount each time, depending on what volume you want to make. So for example, if you wanted to make of it, it would require this much out of that 400 to 500 microliters, to give you 1 ml per 1 million beads per ml. So keeping track of these volumes can be a little cumbersome. So in this scenario, what we can do is use this second spreadsheet here. So let's take these same preparations of protein here and transfer them over to this spreadsheet to see how to use this spreadsheet here. And one thing I didn't point out about these spreadsheets, and that is that on all of these spreadsheets, the green cells is where you're going to enter text or numerical data in, and you don't want to edit the blue cells or the pink cells. The blue cells give you some information about calculations that help you determine whether or not things are being calculated correctly. And the pink cells kind of tell you what volumes you're going to need to pipette. That's the same for all these workbooks that we put out for doing these calculations. So in any event, in this scenario, unlike this one here where you can determine the volume you want, with this tab here, again, you're going to put in the counts that your cell counter or your particle counter gave you. It's going to calculate the actual concentration, and it'll actually count the actual concentration when you put in the right dilution factor, which we don't have there. And then, again, from the scenario we're talking about, we coupled 4 million beads, we brought them up in 500 microliters, and we took out 10 microliters to do our counting in the cell counter. So all these should now have 490 microliters left in them. So now in this scenario, we have this volume left from the coupled stock after we've done our counting. We want them all to be 1 million beads per ml. And so what this spreadsheet tells us is that to each of these preparations, we add this much volume. And when you add this much volume to that remaining in the tube, it'll make them all 1 million per ml, but they're all going to have slightly different total concentrations, as you can see here. But once they're all at 1 million per ml, they're now ready to take over to the next spreadsheet, which tells you how to make the multiplex bead mix, whether you have one bead preparation to add to it to make a singleplex or up to 500 bead regions. So with that, we can look at the next spreadsheet workbook for doing multiplex bead mix calculations. So this spreadsheet has a similar format, as I mentioned earlier, with regards to the different color cells. And it also has a little instruction page here, which gives you some basic instructions on how to use the different tabs in the spreadsheet. But as with the other spreadsheets I just showed you, it's the same format where text or numerical values go into the green cells and blue cells give you information on various calculations that you need to keep track of. And then the pink cells are, again, the volumes you need to pipette. So in this spreadsheet here, you can only enter text in the green cells here. The rest of the spreadsheet is locked. But there's information in these various pop-ups on how to use the information and what to enter in the various tabs. This workbook also has 4 tabs here, as you can see. Most users will use these 2 tabs, the individual bead stocks, with either the percent overage or this one here to combine individual bead mixes to make their multiplex mixes. These are used less frequently. These involve having a multiplex bead mix that already has all the beads in it, but at much higher concentration than you may want to use them at. And in very rare situations will you use any of these 2 tabs, but they're there if needed. And they're going to be used in the same way as you'll use these other 2 individual tabs. So most of the time we use this tab here, which allows you to take your multiplex mixes and combine them in as many plexes as you want to with a volume that you want to deliver the beads to each reaction and the number of reactions you're going to want to need to make that use for that multiplex bead mix. So you can enter things on this thing like the date. So we can enter, let's say, today's date. And you can enter the buffer being used, which for most cases will be our sort of PBS TBN buffer, and maybe that's with 1% BSA. Then here in this tab, you can pick a percent overage. So instead of making the bead mix with, let's say you want to make it for 96 reactions, but you know your pipetter keeps some liquid and retains some liquid so that can throw things off. So in theory, some people make 100 reactions instead. So they'll make 4 microliters extra reactions. In some situations, that causes you to waste beads. So by entering a percent overage, that lets you fine tune that overage that you'll need for making a bead prep, say to go into a trough for dispensing. So you can actually conserve beads a little better rather than estimating how many extra reactions to make of the bead mix. So in this case, for some troughs, like your standard sort of 25 ml trough that you dump your bead preparation in to dispense onto a large plate, usually a 10% overage is sufficient. In this cell here, you're going to enter the number of bead regions that will be in your assay. So let's say we're going to make a 5-plex assay. And let's say, like for example, from the spreadsheet we just did, where we made all of our bead stocks 1 million per ml, at 1 million per ml. And again, if you want to know how to enter data, you can go to these little pop-ups here to see what information should be entered in the format. So again, let's work on the assumption that all of our bead preps we diluted to 1 million per ml. And now let's say of all those bead preparations, the one that has the smallest volume maybe is 1 ml. Because some of those bead stocks, like we saw in the previous spreadsheet, if you decide you want to use the whole bead stock and dilute it, say, to 1 million per ml, some of them will have a different volume in them. And sometimes out of those different groups of couple beads, there may be one that instead of having maybe 1 ml total, maybe that one's going to have only, say, 900 microliters. So when you need to use that particular bead with the rest of them, the volume of this bead that's at the lowest volume will set the limit on how many reactions you can actually run. So if you can, it's best to keep track of your bead volume. So when you do have a multiplex mix, keep track of which one has the lowest volume and enter that value in here. So as far as the number of beads to add per reaction, we generally recommend 2,500 beads to add per reaction. And as far as the volume of that bead mix to add, that can vary. Some applications require 10 microliters. A lot of applications, like various proteomic applications, they require 50 microliters of the bead prep. So whatever volume you want to add this multiplex into the reaction, you enter that here. And again, more information is listed here on that value. In this cell here, you're going to enter the number of reactions. So let's say you want to make enough for a 96-well plate. So you just enter 96 here. Once all this information is entered in here, the two frames down here will tell you how to make that bead mix. In this situation, in order to make this bead mix for 96 reactions, where you're going to add 50 microliters to each well, and for each bead region, it's going to give you this many of each bead region in the reaction. This mix is going to be made by taking 264 microliters of each bead mix, combining them together in one tube, which is going to give you a volume of a little over 1 ml. So this volume here tells you what size vessel you're going to need to mix all this in, especially when you add buffer to that, which is going to bring the total volume to a little over 5 mLs. So all these values here are giving you information on the size of the tubes you're going to need to make this bead mix. The other thing it tells you is that you don't need to concentrate, because at this small plex, the volumes required to generate this bead mix don't require concentration. But let's say you're making a higher plex assay, say a 30-plex assay, where again, all your beads are at 1 million per ml. Your smallest bead prep has 900 microliters. And again, you want to add this much of each bead region to the mix in this volume, and you want to do it for 96 reactions. So you can see here that again, you're still going to be using 264 microliters of each bead prep. However, this time when you're combining 30 of them together, it's going to require or create a total volume of a little over 8 mLs, which you have to concentrate down to a little over 5 mLs. The same volume you needed over here when you were making the 5-plex mix. So by following these instructions now, when you've done this, you'll actually have an accurate dilution of all your beads, so that when you add your 50 microliters to each reaction, you're going to get your 2,500 per reaction, as well as being accurate compared to trying to do this by just having a standard dilution, where some people have done, they say all their beads are at 50 beads per microliter. And they just keep combining them, whether they got a 5-plex or a 30-plex, and you can see how that would tremendously cause a dilution of your beads, because you wouldn't take it through a concentration step if you use that protocol as written in the cookbook. The other thing this calculation can tell you is when you have enough beads to actually use to make your bead preparation. So let's say, for example, you wanted to make 200 reactions. Okay, if you wanted to use these beads to make 200 reactions, you have enough. Let's say let's bump it up to 500 reactions. Okay, so you can see here that if you wanted to make 500 reactions with this bead mix, you wouldn't be able to do it, because that one bead preparation that had the lowest volume is sort of limiting how many reactions you can actually make. So let's say if this was up to 1.5 mLs, that was the lowest volume, then yes, you would have enough to do it. But in essence, if we go back to the 900, what this is telling you is that, okay, you cannot perform this experiment or make this prep for 500 reactions. So you would either have to make more beads with regard to whichever one is limiting, or you'd have to scale back the number of reactions you can run, with these bead preparations that you're currently using. Okay, so somewhere around between 300 and 400 is the number of reactions maximum you could get with this 30-plex based on this concentration, and one of them having a minimum volume of, say, 900 microliters. So these other tabs work the same way. So for example, if we took this information here, and if you wanted to use this tab instead, throw in here, and again, the only difference here is that instead of having that percent overage, you now have to just go by number of reactions to determine how to make your multiplex bead mix. So that's how to use your bead mix calculator. And when you use this calculator, you can also print these tabs out on a sheet of paper landscape and bring them to the bench, so you can have this information at the bench when needed to make these bead mixes. So that concludes this presentation on tools for calculating bead concentrations and making multiplex bead mixes. If you have time, you should also check out some of the other presentations about precision coupling of molar amounts of protein or peptide, as well as doing calculations for coupling or binding to Avidin beads with microgram amounts of protein and peptide. So thank you for viewing this presentation, and good luck with making your Luminex assay. If you need any help, feel free to reach out to your FAS or technical support.